Cells can be divided into two main groups, namely eukaryotes and prokaryotes. Prokaryotes are classified under the monera kingdom and include bacteria. Certain structures are always present in prokaryotic cells, and others are not always observed. The cell wall is always present and is made up of a peptidoglycan called murein. We then have the cell membrane, which forms a semi-permeable barrier. In prokaryotes, the cell membrane can also form infoldings that can perform nitrogen fixation or act as a photosynthetic membrane, which is important as prokaryotes do not have membrane-bound organelles like mitochondria. Structures like the flagellum and the capsule can be seen in some strains of E. coli and is responsible for motility and protection, respectively. Pathogenic E. coli also possess pili to attach to cells, but can also be used in sexual reproduction. They have a circular piece of DNA that carries their genetic material. Some even have smaller pieces of circular DNA called plasmids. Prokaryotes do, however, have ribosomes. These 70-ish ribosomes and all other organelles are contained within the cytoplasm. Let's look at an example. This is an electron photomicrograph of one strain of E. coli undergoing cell division. Prokaryotes are unicellular and about 1 to 5 micrometers in diameter. Using an electron microscope, we can observe the internal structures of the cell. We can see that there are circular DNA present in both cells. It is not as compact as a nucleus and will appear lighter. We can also identify the cell wall. The interior of the cell has a grainy appearance, which is indicative of ribosomes floating in the cytoplasm. Next, we have a pathogenic strain of E. coli attaching to the epithelial cells in the small intestine. Here we can see that the prokaryotic cells are much smaller than the eukaryotic cells. There is no cell organelles visible, but the absence of microvilli on the epithelial cells are evident of the E. coli being pathogenic and causing destruction to the lining of the small intestine. Eukaryotes make up the other four kingdoms. These are animals, plants, fungi, and protists. These four kingdoms have several things in common, namely a cell membrane that is semi-permeable and surrounds the cytoplasm, a nucleus made up of a nuclear membrane, nucleolus, and chromatin. The chromatin can either be condensed into a chromosome form or spread out through the nucleus. Continuous with the nuclear membrane is the endoplasmic reticulum. Usually, this endoplasmic reticulum is a rough endoplasmic reticulum. All eukaryotic cells also possess ADS ribosome for protein synthesis in their cytoplasm, mitochondria for cellular respiration, and a Golgi body with vesicles for the packaging and transport of molecules. These are usually for the exocytosis of hormones. Eukaryotes also possess lysosomes, which are called dendritic vacuoles in plants and in fungi. All these organelles are compartmentalized and held into place by the eukaryotic cytoskeleton. The first thing that sets animals apart from other three kingdoms is the fact that they do not possess a cell wall. The cell wall is always present in fungi as chitin and in plants as cellulose, but it's optional for protists, and if it is present, it's present as cellulose or other types of polysaccharides. Plants and protists can also contain chloroplasts for photosynthesis. Large vacuoles are also present in fungi, plants, and protists. In protists specifically, these are specialized vacuoles called contractile vacuoles, which are responsible for removing water from the cell. This is important as some protists do not have cell walls to protect them from osmosis and water moving into the cell. The connection between protists and animals comes from a shared presence of microvilli, flagella, and cilia. Microvilli is important for the increase in surface area that aids in absorption, especially in digestion. Flagella and cilia are used for motility, and cilia can also be used for the attachment onto other cells. When you're trying to identify a eukaryotic cell, keep this in mind. If the cell has no cell wall and no chloroplast, it's an animal cell. If the cell has a cell wall with chloroplast, it's a plant cell. If the cell has a cell wall but no chloroplast, it's a fungal cell. And if the cell has a contractile vacuole, it will be a protocyst. An example of an animal cell is a monocyte, which forms part of the human immune system. In this photomicrograph, we can see a large irregularly shaped nucleus. It's pressed against the cell membrane by an erythrocyte that is being digested. We can also see the cell membrane with the absence of a cell wall. You can see the cytoplasm and no other cell organelles. Next, we have a plant cell, more specifically a parenchyma cell. The cell wall is prominent in this photomicrograph, so too is the very large vacuole pressed against the cell wall. We then also see a large nucleus that consists of the nuclear membrane and the nucleolus. This small organelle is a mitochondria, and several are visible in the cytoplasm of the cell. The big identifier is the large chloroplast that will set the plant cell apart from fungal cells. In comparison, here we have a fungal cell. The cell wall, cytoplasm, and nucleus is visible, but the absence of chloroplast indicates that it is a fungal cell. On the side of the cell, we can also see the nucleus, which is indicative on a eukaryotic cell. Lastly, we have a photomicrograph of a ciliate, which falls under the kingdom protocyst. The nucleus is very large and prominent, and the chromatin can be seen inside. No nucleolus can really be identified in this example. It also possesses a large vacuole and several smaller contractile vacuoles that are closer to the cell membrane, enabling them to push water out of the cell. Smaller organelles like mitochondria can also be seen in this example. On the outside of the cell membrane, we can also see cilia, which will help with motility and attachment of the cell. Viruses are technically not living and will not fall under any of the 5 kingdoms, and neither eukaryotic or prokaryotic. Instead of a cell membrane, they possess a protein coat called a capsid. The capsid has antigen molecules attached that can identify the cell. Some viruses do, however, possess an envelope that's made up of phospholipids. They also have a self-replicating piece of DNA or RNA, which will be incorporated into the host's genetic material to replicate in a form of asexual reproduction. Another well-known form of virus is the bacteriophage. This type of virus will not enter a host cell and would rather inject its genetic material into the host. The host cell will then replicate the viral particles, and once the viral particles are assembled, they will lyse the cell and spread to other cells. The first photomicrograph shows HIV particles infecting a lymphocyte. The lymphocyte, being eukaryotic, is much larger than the viruses, which are usually between 20 and 200 nanometers in diameter. There are also no organelles observable inside any of the viruses, unlike in the eukaryotic lymphocyte, where the cytoskeleton is visible. Then we have a transmission electron micrograph of the bacteriophage T4. It is easily recognizable by its icosahedral head and spiral body with tail fibers. On the right is a more magnified photo where the self-replicating piece of DNA can clearly be seen inside the icosahedral head.